Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and hello everyone. My name is Nurul Farhana binti Swahili. I am now currently studying Diploma in Science at UiTM Tapa. So today in this video, I will share with you guys a few things about aerobic respiration and fermentation. What we will discuss in this video. Alright, in this video, we will discuss about what is aerobic respiration and fermentation. Next, the difference between aerobic respiration and fermentation, and why do organisms undergo fermentation. So, what is aerobic respiration? Aerobic respiration is a process of cellular respiration that takes place in the presence of oxygen gas to produce energy from food. Aerobic respiration occurs in all multicellular living organisms such as animals, plants, and other living things. All living organisms will undergo aerobic respiration because we all need an energy. The steps that involve in aerobic respiration is glycolysis, formation of acetyl coenzyme A, citric acid cycle, and electron transport chain. So in this process, aerobic respiration, we will require glucose and oxygen to produce carbon dioxide, water, and energy, which is in form of ATP. So this is aerobic respiration diagram. Aerobic respiration diagram given in this slide represent the entire process of aerobic respiration. The different cycles involved in aerobic respiration, such as glycolysis, Krebs cycles, electron transport chain. Okay, so the first step is glycolysis. Glycolysis is a primary step of aerobic respiration and takes place within the cytosol of the cell. Okay, so in glycolysis, there are two phases that involves. The first one is investment phase and the second one is payoff phase. In investment phase, ATP is being utilized, while in payoff phase, ATP is being built up. So in investment phase, glucose will undergo a few steps. The first one is glucose will be converted to glucose 6-phosphate, and the enzyme that involved is exokinase. And then glucose 6-phosphate will be converted to fructose 6-phosphate, and the enzyme that involved is phosphoglucosoisomerase. After that, fructose 6-phosphate will be converted to fructose 1-bisphosphate, and the enzyme that involved is phosphofructokinase. Then the final one is fructose 1,6-bisphosphate will be split into two. The first one is dehydroxyacetone phosphate, and the second one is glyceraldehyde 3-phosphate. The enzyme that involved is idolase. Alright, so dehydroxyacetone phosphate and glyceraldehyde 3-phosphate will undergo payoff phase and will be converted to 1,3-bisphosphoglycerate, and then it will be converted to 3-phosphoglycerate, and then will be converted to 2-phosphoglycerate, and will be converted to phosphoenol pyruvate, and finally they will be converted to pyruvate. Then we will get 2-pyruvate as a final product of glycolysis. During this glycolysis process, the glucose molecules are splitting and will produce 2 ATP and 2 NADH molecules, which are later will be used in the process of aerobic respiration. Next, after glycolysis, we move to the formation of acetyl coenzyme A, the second step in which is the second step in aerobic respiration. In this process, pyruvate is oxidized in the mitochondria and 2-carbon-acetyl group is produced. The newly produced 2-carbon-acetyl group binds with coenzyme A, producing acetyl coenzyme A. After that, we go to the citric acid cycle. The third step in aerobic respiration is the citric acid cycle, which is also called the Krebs cycle. In this stage of aerobic respiration, the oxaloacetate combines with the acetyl coenzyme A and produce citric acid. In this cycle, a series of reactions that give off CO2 and produce 1 ATP per cycle. Each turn of the Krebs cycle, 3 NADH, 1 FADH, and 2 CO2. because we have two pyruvate, so the final we will get 2 turn of to produce at 6 NADH, 2 FADH, 4 CO2 and 2 ATP. Next is electron transport chain. This is the last step in aerobic respiration. In this phase, the large amount of ATP molecules are produced by transferring the electrons from NADH and FADH. It occurs in cristae, which is an inner membrane of mitochondria. The movement of electron along the electron transport chain will contribute to chemiosmosis and ATP synthesis. A single molecule of glucose creates a total of 34 ATP molecules. So that is all about aerobic respiration. We can conclude that aerobic respiration is the process of utilization of oxygen to break down glucose to produce ATP. The pyruvate is then converted into acetyl-CoA in the mitochondrial matrix. Then the Krebs cycle occurs twice per glucose molecule because glycolysis will produce 2 pyruvate. Next, the protein complexes are arranged on the inner mitochondrial matrix so that the electrons pass from one reactive molecule to the other. This is known as the electron transport chain. And the last one, ATP synthase produce ATP from ADP and inorganic phosphate. Alright, so our next discussion is about fermentation. Fermentation is or we can call it as anaerobic respiration. Is a cellular respiration that takes place with the absence of oxygen. Anaerobic process such as fermentation take place inside a variety of cells, including yeast cell. The example is lactic acid fermentation. In yeast, there is incomplete oxidation of glucose that happens in an aerobic environment. Here, pyruvic acid is converted into carbon dioxide and ethanol. Some bacteria produce lactic acid from pyruvic acid by means of lactic acid fermentation. So this is a lactic acid fermentation diagram. Steps that involve is glycolysis and NAD plus regeneration. In this diagram, we can show that the glucose is broken down into glyceraldehyde 3-phosphate and then to 3-phosphoglyceric acid. During this, NAD plus is converted into NADH. The 3-phosphoglyceric acid from the 3-phosphoglyceric acid forms phosphophenylpyruvic acid, which later forms the pyruvic acid. Net 2 ATP molecules are formed in glycolysis. This pyruvic acid is reduced to lactic acid with the help of reducing agent NADH, which reoxidizes to NAD+. This process produces two lactic acid molecules from 2 pyruvic acid molecules. This reaction happens in the presence of enzyme lactate dehydrogenase. Lactic acid fermentation is the best method used for food preservation. Lactobacillus is the most commercially used bacteria for this process. They are used in production of pickles, sour beer, fermented fish, and yogurt. So that is all about lactic acid fermentation. Next, we move to the aerobic respiration and anaerobic respiration. The difference between these two. In aerobic respiration, oxygen is required. 38 ATP produced per mole of glucose used. Stage that involve is glycolysis, link reaction, Krebs cycle, electron transport chain. This aerobic respiration occurs in eukaryotes. It's lower rate of ATP production and can be found in cytoplasma and mitochondria. But in anaerobic respiration, oxygen is not required. 2 ATP produced per mole of glucose used. Stage that involve is glycolysis and fermentation, occurs in eukaryotes and prokaryotes, rapid ATP production and can be found in cytoplasm. So the question that always been asked is why the organism undergo fermentation. So the answer is, as we all know, fermentation is the process in order to form energy in the absence of oxygen. All the organisms who have no oxygen will undergo anaerobic respiration, which involve 2 steps, which are glycolysis and fermentation. So this is a few reference that I use in order to complete this video. So that's all from me. I hope whoever listened to this video can at least get some input. Thank you for listening.